Can somebody please confirm you can hear me?
Yes.
Thank you. Sorry for being late. They changed the way I sign into the computer, and I had to download an app on my phone and install it and run it and sign into my account through the app, and it was just all a mess. Before we begin, are there any questions besides why the heck I was so late? Come on, open. All right, let me share my screen here. Should be seeing my screen now. All right, today is June 17th, and we should be finishing chapter 2 and starting chapter 3 today. We have a lab today. It'll be lab 3 on microscope basics. And hopefully you read the textbook on chapter 3 through the end of the section on compound light microscopy. You need to do that for lab 3 and lab 4. Any questions about what we're going to do today?
No.
All right, great. So I have that we were talking about amino acids last time and we had covered this slide. That there are about 20 different amino acids to most organisms. Humans have exactly 20. Amino acids are linked together by a peptide bond. Peptide bonds are kind of unique, not only in living organisms, but in the universe. A peptide bond is a link between a nitrogen and a carbon. And that bond is not very common in the universe. It does happen, however, in peptides, why it's called the peptide bond. What happens is you have two amino acids which come together. Water is pulled out, the OH is pulled out of one amino acid, the hydrogen out of the other amino acid, and then that nitrogen links directly to that carbon in a dehydration synthesis reaction, forming the peptide bond and water. A polypeptide is just a chain of different amino acids linked together by peptide bonds. Uh, when the protein is growing, meaning when it's a few amino acids, I'm not sure what the smallest number of amino acids can be, and you still call it a peptide, but it's a few amino acids. Oops, didn't mean to advance that. So a few amino acids is a peptide, many amino acids is a polypeptide. And as the protein is growing, when the last amino acid is added, that's when we call it a protein. Before it is its final product, the protein, you can call it a growing protein, or you can call it a polypeptide. But now once the last amino acid is added to that polypeptide, we then call it a protein. Any question about any of that? All right, proteins have four levels to their structure. There's the primary level, the secondary level, the tertiary level, and the quaternary level. Let's watch a little movie talking about the four levels of protein structure.
Grammarly is an AI writing partner that works seamlessly with your everyday apps and helps professionals write better and faster so they can get all of their work done.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid.
All right, let's skip
So an
this
amino acid has
and then get on to the levels of protein.
two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the of protein structure,
Let's go back a little.
bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds.
All right, I want
The
to
NH group of one amino acid.
stop right there. This video is only talking about one interaction between amino acids which stabilize the structure of the secondary structure of the protein, the hydrogen bonds. There are other interactions between amino acids which stabilize the secondary structure, and I'll talk about them because this video is not talking about them.
Interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about this video? I'll leave it up in case I want to bring up something, but I don't think I will.
So for the secondary structure,
Uh-huh.
That would include like hydrogen bonding, London dispersion forces, and like disulfide bonds too, right?
Disulfide bonds do not occur in the secondary structure. They occur
Okay.
only in the tertiary structure,
Okay. Thank you.
right? So the primary structure is just the specific sequence of amino acids in a polypeptide chain. So you got the orange amino acid, followed by the blue amino acid, followed by the yellow amino acid. And it's just the sequence of amino acids uh, derive the primary structure. So when the protein is first made, all it has is the primary structure. The amino acids are not interacting with each other yet, and so all you have is the primary structure of the protein. But once the protein is made, the nearby amino acids start interacting with each other. These are localized interactions between neighboring or nearby amino acids. And the video did tell you two of the secondary structures. I probably won't ask you about that, but you can have an alpha helix where it forms a helix like that. You can have the beta pleated sheet where there's it looks like this protein is running this way and then this way and then that way, and it forms links between the amino acids here. And that would be a trying to figure out, is that the protein there or is that the protein there? I'm not sure. There are interactions like hydrogen bonds between the amino acids in the beta pleated sheet. The beta pleated sheet has in the video, maybe there's a better video of it. Let me go back to that. No, that's not it. Sorry. This would be the alpha helix, and where the protein is sort of folding on each other and then forms hydrogen bonds and other links between the two folds. This would be a beta pleated sheet. And you didn't mention the third structure is a random coil where the protein just takes a random type shape. And so there's actually three structures to the protein, the alpha helix, the beta pleated sheet, and then the random coil. So the secondary structure forms from the localized interactions between neighboring amino acids. These localized interactions can be forces of attraction and they can be forces of repulsion. Now, the video mentioned the hydrogen bond forming between two amino acids. That is a force of attraction, where one amino acid hydrogen bonds to another. Another force of attraction is the hydrophobic interactions, where a nonpolar amino acid likes to be together with another nonpolar amino acid. And these forces of interaction or attraction are very weak. They're called van der Waal forces. But you've actually seen van der Waal forces hold the molecules of oil together. Like when you have your salad dressing and you have your oil there, the oil is a liquid. And what's holding those oil molecules together are simply because the molecules are nonpolar. Nonpolar molecules are attracted to other nonpolar molecules by van der Waal forces, and these forces make the oil molecules come together and form a liquid. That's why the oil is a liquid, because the molecules, they're not solid, where the van der Waal forces break and reform all the time, so the oil molecules flow over each other, forming a liquid. If the oil molecules were not attracted to each other, you'd have gaseous oil, and you would not have a liquid oil. And if the oil molecules were ever to not break the forces of attraction between them, you would not have an oil. You'd have solid oil. Like when you put olive oil in the refrigerator or in the freezer, it will solidify, and the van der Waals forces of attraction will hold the oil molecules together, forming the solid oil. Any question about any of that? Anyways, that's the nonpolar forces of attraction. They're called van der Waals forces if you had that in your preliminary biology class. You also have forces of attraction where, let's go with this amino acid and that amino acid. If this amino acid has a negative charge and this amino acid has a positive charge, they can form an ionic bond between them. And that will be a strong force of attraction. On the other hand, you can have forces of repulsion. If this has a positive charge, this amino acid, and that amino acid has a positive charge, then that would be a force of repulsion. These two amino acids will not come near each other because they both have a positive charge. And that's a +1 positive charge, which is an ionic charge, not a polar charge. A polar charge is a partial positive and a partial negative, forming the hydrogen bond between two nonpolar amino acids. Now, have I discussed all of them? Ionic bonds, hydrophobic interactions. Oh, there can be a force of repulsion, and that is if this is a polar amino acid and that's a nonpolar amino acid, these two amino acids will be repulsed from each other. So both forces of attraction and forces of repulsion can give the protein the secondary structure, where the localized amino acids start interacting with each other, giving the rise to the secondary structure of the protein. Now, the key to note is only the nearby amino acids interact with each other to give the secondary structure. The tertiary structure is where the amino acids from all over the protein start interacting with each other, having forces of attraction and forces of repulsion. All of them that we saw in the secondary structure occur in the tertiary structure. The difference is they don't have to be local interactions between amino acids. Like this is an amino acid at the end of the protein, and it's interacting force of attraction with a amino acid near the beginning of the protein. You can also have forces of repulsion, like maybe right here and here. These two amino acids are being repulsed from each other. In the tertiary structure, we have one other force of attraction that is not seen in the secondary structure, and that is a disulfide bond. Blow this up. The amino acid cysteine has a sulfhydryl group. And let me shut that down, and I'll try and draw it. So we have, hopefully this is right. We have a cysteine here and a cysteine here. And let me see if I can do that. Oop. Ah, there we go. which I guess I should put it on this side. Attached to that cysteine is a sulfhydryl group. I'm just going to draw it. Nope, that's not what I want. Just going to draw it in. So there's a sulfhydryl group here. That's an S. A sloppy S, but you're getting the picture. And attached to that's a hydrogen. Attached here is a hydrogen. Oop. Kind of getting it. All right, well, that hydrogen can come off. It'll just go into solution, be hydrogen ions in solution. And this one can come off as well. And when they come off, the sulfurs can then form a link between them, which is a covalent chemical bond between the two sulfurs. This is a very strong bond because it's a covalent bond. It's not a hydrogen bond. An example of a link in your sulfur is when you perm your hair. What the perming does is create a link between the cysteine in your hair, giving you the perm. And this will be a very strong bond. It'll last about a month of showering. Whereas my mother used to put her hair in curls, put on the curling hat, and plug in the curling machine. Don't ask me what it was called. I'm just going to call it a curling machine. And then it would put steam in the hat. And then she would do this for about 10 minutes, take the hat off, take off the curls, meaning whatever the bobby pin was and the curl. And then her hair would have this curl. And then she would go she lived in Western Oregon, in the Western Oregon weather, and there'd be frequently mist in the weather. And that hydrogen or the water in the mist would get in her curl, and then the curl would sag, showing you how weak a hydrogen bond is. It can't survive a little bit of water, just the mist in the air in Western Oregon or Washington. Whereas the perm forms this sulfide dye bridge between the two cysteines. And this is very strong, and it lasts about a month of regular showering. Any question about any of that? Anyways, this is the force of attraction happening only between cysteines, the amino acid cysteine, in the tertiary structure. Any questions about any of that? So all of the forces of attraction and repulsion that we saw in the secondary structure, you also have in the tertiary structure. The tertiary structure also has the disulfide bonds between two cysteines, which make a very strong force of attraction between this cysteine here and that cysteine there. Any question about any of that? All right, some proteins, all they ever have will be the primary structure, the secondary structure, up to the tertiary structure. They never get higher than the tertiary structure, and that's because the protein only has one subunit. Only has one polypeptide, which makes up the protein. But there are some proteins which are made up of more than one subunit, such as an antibody or hemoglobin, where 4 polypeptide subunits come together to make the final protein structure. And these will have the same forces of attraction we saw in the tertiary structure, where this unit then reacts with that protein unit, which then reacts with this protein unit and possibly with that protein unit. And there will be forces of attraction between amino acids on this polypeptide and that one, as well as with this one, and then with that one. It looks like there's a force of attraction between these amino acids, maybe a force of repulsion between this amino acid and that one. So many proteins only have up to a tertiary structure, which would include every enzyme I know of. Only have a tertiary structure. And then only a few proteins have quaternary structures, such as hemoglobin, insulin, and antibodies. Hemoglobin and antibodies have 4 polypeptides that come together to make the final protein, the quaternary structure of the protein. Any question about any of that? All right. Proteins can be globular, where their shape is roughly spherical, they're compact. Hemoglobin is an example. Most enzymes are also globular. And then the protein can be fibrous, that's thread-like. Many of the structural proteins, like collagen, are fibrous proteins. Proteins can also combine with other molecules to make conjugated proteins, the combinations of amino acid with other organic or inorganic components. These conjugated proteins are usually named by their non-protein component, like we call glycoproteins. This is a protein linked to a glycogen or whatever. And a glycogen is a carbohydrate. So we have a carbohydrate moiety linked to this protein. And then there's a lipoprotein, which is, of course, a lipid linked to a protein. Proteins can also denature, and that is when a protein loses its three-dimensional shape, where it breaks hydrogen bonds that should be there, or it forms hydrogen bonds which should not be there. You can have the protein lose its three-dimensional shape and take on another shape. And that will cause the protein to lose its function, and that we call protein denaturation. When a protein is partially denatured, the protein has some function, but it's not functioning at its optimal level, or 100%. And then when the protein is no longer functional, is functioning at 0%, we call it totally denatured. You can get the protein to denature by putting it in the wrong temperature, the incorrect pH, or the incorrect salt concentration. Like our enzymes work best around 37 degrees. We're talking about an internal enzyme. And they work best around pH 7 to 7.4. And whatever the blood salt concentration is, I don't know, about 0.09%. If you put it at a different temperature, the protein will begin to denature and will start losing its functionality. And when you put it at an extreme temperature, different from the optimal temperature, the protein will have no function, will be totally denatured, such as when you boil the protein or you put the protein in the refrigerator, or enzymes cannot work in the refrigerator. And if you put the enzyme in, like pickle juice, for example, our enzyme will no longer function. It'll totally lose its three-dimensional shape and will no longer have its functioning. And I'm trying to remember, pickle juice, I think, is around pH 4. And salt concentration, you take it off from 0.09% sodium chloride, and the protein will start to denature. Any questions about any of that?
Are you able to show slide 41 again really quick?
Sure.
Thank you. Okay, perfect.
All right. All right. Any questions about proteins? If not, let's move on to nucleic acids. There are two
I had
unique.
a question real quick.
Uh-huh.
So, for like the protein denaturing, when you get like a disease like mad cow disease and your proteins stop functioning, is that due to denaturing or is that a different process?
That is a different process. We will talk briefly about, I guess, mad cow disease, but the prion comes up to the normal functioning protein in the nerve cell and then gets that protein to take on a different folding pattern. And the normal protein, which is needed by the nerve cell, takes on the folding pattern of the prion protein. And now you got two prion proteins in the cell, whereas before you only had one prion protein in the cell. And that's a little bit of a complicated story, but when we talk about prions, you'll learn that the prion is a different folding pattern to a protein which is in the nerve cell. But in the nerve cell, the protein has a different three-dimensional shape, and then that protein is needed by the nerve cell for the nerve cell to function.
Okay,
Okay?
thank you.
All right. Nucleic acids, there's two of them you need to know about, and that's DNA and RNA. DNA, another name for it is deoxyribonucleic acid. RNA, another name for it is ribonucleic acid. Nucleic acid consists of monomers called nucleotides. A nucleotide itself consists of three parts. There's a 5-pentose sugar. Let me blow this up. So there's the sugar. And in DNA, that is deoxyribose. In RNA, not shown here, it would be ribose. But that's the 5-carbon pentose sugar. And then a phosphate group shown here. And then a nitrogen-containing base shown here. There are two types of nitrogen-containing bases, a purine or a pyrimidine. Let me end the show there. And there we have the nucleotide containing the nitrogen-containing base, the 5-carbon pentose sugar, and then the phosphate group. Let's go on and talk about the different nucleotides. There are 4 nucleotides in DNA, 2 purines and 2 pyrimidines. The purines are adenine or A, and you can learn either name, adenine or A, guanine or G, and then there are 2 pyrimidines in DNA, cytosine or C, and thymine or T. How you can recognize what's a pyrimidine from a purine is pyrimidines has a Y in it, and all of the pyrimidines in DNA have a Y in the name. Cytosine and thymine have a Y. All of the purines do not have a Y in the name. Any question about any of that? Now, for RNA, there are also 4 different nucleotides. There is adenine, there is guanine, and there is cytosine. In RNA, there is not thymine. Thymine is replaced by uracil. And how I remember uracil as a pyrimidine is uracil replaces thymine, and thymine has the Y in it. So uracil is also pyrimidine. Generally, I don't talk much about RNA, but if I do, the 2 pyrimidines would be cytosine and uracil. The 2 purines would be adenine and guanine. The pyrimidines and the purines are important because the DNA binds to each other by hydrogen bonds so that the bases are complementary to each other, meaning an adenine always pairs with the thymine in DNA we're talking about. Adenine always pairs with thymine, so the purine adenine pairs with the pyrimidine thymine. And let's see, blow this up. And the A and the T have two hydrogen bonds between them, forming the bonding between one strand of DNA and the other strand of DNA, meaning two strands of DNA are only held together by the hydrogen bonds between the complementary bases. And then the other complementary base pair is C and G. Guanine only pairs with cytosine, or cytosine only pairs with guanine. Doesn't matter how you say it, but and then the CG have three hydrogen bonds between them. The AT have two hydrogen bonds. And let me see if I can show you. There's a CG showing three hydrogen bonds linking this nucleotide to that nucleotide. So DNA, the sugar is deoxyribose. It is double-stranded, the molecule. At least most of the time in the cell, it is double-stranded. And one strand is held together by the sugar phosphate backbone. Phosphate is in the pinkish color here. There we have the nucleotide. There we have the blue right there being linked to the phosphate. And then on the other side of that sugar, the 5-pentose sugar, which is deoxyribose, we have, which isn't shown well. Let's go up here. This one is linked to this sugar here, the blue, and then there is the phosphate there. And so this one strand is held together by covalent chemical bond, phosphate covalently linking to the sugar, covalently linking to the phosphate. You'll note that the nitrogen containing bases are not holding the single strand of DNA together. They're out on the spiral staircase, forming the steps of the spiral staircase. And then the other strand of DNA is only held together by the hydrogen bonds between the complementary pairing, meaning the guanine always pairs with cytosine, and that nitrogen-containing base here and this nitrogen-containing base here, as well as the hydrogen bonds, form the step of the spiral staircase of the twisted ladder of DNA. Any question about any of that? All right. Let me see if I've got anything I'm going to say here. In DNA, the bases are only A, T, C, and G. In RNA, the sugar is ribose. Ribose has one more oxygen than deoxyribose. Let me see if I can show deoxyribose. So here's deoxyribose. It has only one OH group on it. Ribose has two OH groups. You don't need to know that, but this is ribose. And once again, the RNA is hooked together by the sugar phosphate backbone, meaning we have a phosphate here, which links to the ribose. And then this ribose will link to a phosphate here. And this will make the RNA hooked together, phosphate, ribose, being a covalent chemical bond making the single strand of RNA. The nucleotide base is off on the side here. It doesn't link the RNA together. There are different bases on the RNA. It is adenine or A, guanine or G, cytosine or C, and then uracil or U. RNA is a single-stranded molecule. At least in our cells, RNA is a single-stranded molecule. We only have one single strand of RNA. Any questions about any of that? There are three major kinds of RNA in cells. There's transfer RNA or tRNA, there's messenger RNA or messenger RNA, and there's ribosomal RNA or rRNA. All of these three forms of RNA are important in protein translation. The messenger RNA brings the code for the protein to the ribosome. The tRNA reads the messenger RNA and attaches the correct amino acid for the code on the messenger RNA. And then the rRNA, the ribosomal RNA, gets together with the ribosomal protein to make the ribosome. Any questions about any of that? All right, let's move on to ATP. ATP stands for adenosine triphosphate. ATP is the primary energy source for cellular activities. Usually, if a cell wants energy for something, it gets that energy from ATP most of the time. ATP has ribose in it. It has adenine in it. So that's the nitrogen-containing base. And then it has 3 phosphate groups in it. You'll note that the third phosphate group and the second phosphate group is attached with a squiggly line. These squiggly lines are high energy bonds, and if the cell wants energy for some purpose, it will break this phosphate off, which will release a lot of energy, and then the cell can use some of that energy for its energy needs. I'm saying some of that energy because, as you know, whenever energy is changed from one form to another, some of that energy is given off as heat, and the expression is usually some of that energy in this ATP molecule will be lost as heat. So only some of that energy given off when this comes off will be energy that the cell can use. Some of it will be converted into heat. If the cell needs more energy than just this one phosphate group can give, the cell can then take off this phosphate group, and that also releases a high amount of energy, which the cell can use for its energy needs. Any question about any of that? All right, because the cell usually gets the energy it needs from ATP, ATP is called the primary energy source or the currency or energy currency of cells, meaning whenever the cell needs energy, it usually gets it from ATP. And why we call it currency is because whenever you need to buy something, you usually use currency. In the old days, that's the only way you could buy something unless you traded for it. Obviously, now you've got Bitcoin and all other types of funds, but it all relates to currency. ATP, adenosine triphosphate, is made by dehydration synthesis. You can get ADP, adenosine diphosphate, get together with an inorganic phosphate group, and you need energy coming into the system. And then you can get the reaction coming this way, where this phosphate group will bind to ADP, forming ATP and water. And then when the cell needs energy, it will add water and then break the ATP by a hydrolysis reaction, releasing the third phosphate, and that releases a large amount of energy. Some of that energy can be used for the cell's energy needs. Any question about any of that? So I've got a question for you, since nobody's asking a question. Where do we find ATP? What cells need a lot of energy for the cell to work? Actually, all cells need energy, but what cells need a lot of energy when they're working?
The heart.
Yeah, the heart muscle. Actually, any muscle, while the muscle is working, needs a lot of energy. And we get a lot of that energy for the cell's work from ATP, meaning you break it down, releasing the energy. All right. Hydrogen bonds are molecules that we have discussed in this chapter. So what molecules have we discussed that have hydrogen bonds that give the molecule an important property? What molecules have we discussed that have hydrogen bonds? Come on, we've discussed a lot of them.
Proteins.
Proteins, yes, they can form hydrogen bonds between them. What's another molecule? You don't get the...
Is a peptide bond is not one, isn't?
No, a peptide bond is a covalent bond
Oh, that's a
linking
stronger one. Okay.
the protein together, or the amino acids together to form the protein. So the peptide bond is not a hydrogen bond. So what's another molecule that has a
Water.
hydrogen bond? Say again, I didn't hear you.
Water.
Yeah, water has hydrogen bonds. Without hydrogen bonds, we would not have liquid water. And then the third molecule we've talked about that has hydrogen bonds, which are very important to the molecule, is DNA. If you didn't have hydrogen bonds between two single strands of DNA, you do not have double-strand DNA. We would only have single-strand DNA.
Is oxygen one?
Let me think about that. Oxygen gas does not form hydrogen bonds, mainly because there's no hydrogen in oxygen gas. Like in water, there is hydrogen and oxygen, and you can get the hydrogen of one oxygen molecule linking to an oxygen of another water molecule, forming the hydrogen bond. But in oxygen gas, there is no hydrogen bonds. All right, what would happen to these molecules if all the hydrogen bonds were to suddenly disappear in the universe? Would we have double-strand DNA?
No.
No. Would we have liquid water?
No.
No. So we would not have all of these important molecules and properties to these molecules if all the hydrogen bonds were to suddenly disappear. And that's my point, of that hydrogen bonds are extremely important, not only to us, but to the universe. Any questions about any of this chapter? If not, I'm going to begin chapter 3. Excuse me. And close this down. I guess I can close that down too. Thank you guys for sharing your answers. I know it's not the most comfortable thing to do, but the worst you're going to be is wrong. And I'll just say, guess again. Chapter 3, Observing Microorganisms. This slide shows you the major goals and the rough outline, which I almost always show you in the first slide of the chapter. The major goals and rough outline, know the terms of this chapter. What's resolution, refraction? What are stains? What's a basic and acidic dye? Is a smear preparation? What is a mordant? And what is peptidoglycan? 2, understand the sizes of organisms and the properties of microscopes. 2A, understand the major differences between a light microscope and an electron microscope. 3, understand why staining improves resolution. Actually, a very simple thing going on. And then know the 3 types of stains. We have simple stains, differential stains, and special stains. And then 3B, we're going to talk about the Gram stain, understand the Gram stain, its procedure, and the basis of its differential staining. Any questions about what we're going to be covering? All right, when we're observing microorganisms, you need to know that in science and in medicine, we use the metric system as our unit of measurement. You are expected to know the metric system because science and medicine use it. For the metric system, once you get to know it, it's actually quite easy. And that's because everything is based on a power of 10. And then the names repeat for the different units of measurement, meaning a kilo means 1,000 units of the basic unit. So for the meter, we have a kilometer, and that is 1,000 meters. For a gram, that's the basic unit of the weight or mass, and we have a kilogram. So just repeat the prefix, and that would be 1,000 grams. So you should know a kilo, meter, or a kilo. You should know a meter. You should know a centimeter. That is 100, 1/100th of a meter. And it's based on a unit of 10, like there's a decimeter. So this is 1/10 of a meter. And then a centimeter is 1/10 of a decimeter, or 1/100th of a meter. You don't need to know a decimeter. We won't use it. But you will need to know a centimeter. You will need to know a millimeter. And that's 1/10 of a centimeter, or 1/1000th of a meter. There are units of measurement between a millimeter and a micrometer. We're not going to use them. And science and medicine don't tend to use them as well. But we do need to know a micrometer. That is 1/1,000,000th of a meter. And a micrometer you need to know because that's the size that we measure things under a light microscope. And then there's a nanometer. That is 1/1000,000th the size of a meter, meaning there are 1 billion nanometers in a meter. And you kind of need to know a nanometer because that's the size that you use with an electron microscope for measuring the size. You don't need to know a picometer, and I bet you've never even heard of a picometer. And it is, I used to know this name. 1 times 10 to the 12th of these picometers to make a meter. And you don't need to know a picometer. You only need to know the metric units of conversion. So you should know that a kilometer is 1,000 meters. A centimeter is 1/100th of a meter. A micrometer is 1/1,000,000th of a meter. and a nanometer is 1/1000th of a meter. And then convert between them. So if I say something is, I don't know, 0.2 kilometers, then that would be 200 meters long. And usually when I convert between kilometers and let's say centimeters, I'll first convert the kilometers to meter and then convert that number to centimeters. You do not need to know how to convert metric units to the US equivalent, where I used to know this, a meter equals about 1.09 yards, or a centimeter equals about 0.4 inches. Okay, it's actually 0.394 inches, but you don't need to convert between English or US. It's no longer English, US measurement and then metric, or metric and a US measurement. But you do need to know how to convert between metric units. And then for something like water, 1 meter is equal, 1 millimeter is equal to 1 cubic centimeter. So 1 milliliter of water is equal to 1 cubic centimeter of water. So when we're talking about water, the weight is built into the system, and 1 milliliter of water equals 1 cubic centimeter or 1 cc of water. And energy is built into the system as well. 1 British thermal unit can raise 1 gram of water 1 degrees Celsius, and we won't talk about that one. But you should know how to convert between the length as well as grams. You should be able to say this: 1 gram equals, well, let's say 1,000 grams equal 1 kilometer. 1/100th of a gram equals 1 centigram, etc. You just change the ending unit, the meter versus the gram. And what's the other unit? Let's see, gram. Oh, liter. A liter is the volume that you use in the metric system, and we have 1 liter being our base unit for the volume. A kiloliter is 1,000 liters. A centiliter is 1/100th of a liter, etc. Any question about any of that? All right. So you should know most of this stuff for the exam. You don't need to know a decimeter. You don't need to know a picometer, but everything else you should know. And you shouldn't know the US equivalents. Now, here's another reason why you should know the metric system. This map is showing you all the countries in the world that do not use the metric system. And as you notice, the US is the only large country that doesn't use it. All the other countries in the world use it, meaning there's only 3 countries in the world that do not, and 2 of them are converting to use the metric system. The United States is actually one of those which is converting to use the metric system. The US started converting to use the metric system in legislation that Thomas Jefferson signed when he was president. And obviously we're going very, very slowly. But if you look at most cars today, if not all cars today, they will show you the speed you're going in miles per hour as well as kilometers per hour. And then on some signs, it'll show you in big letters that you should go this speedometer, meaning you should go 60 miles per hour. And then in some of them, they'll have in smaller print, you should be going so many kilometers per hour. Anyone notice that? The US is slowly converting to the metric system. I doubt we'll be converted by the time you die, but we are slowly converting to using the metric system. And I don't remember the other country which is converting. But like I said, 2 of the 3 which do not use the metric system are slowly converting to using the metric system. All right. So, some typical sizes of microorganisms. A eukaryote cell, the average size is between 10 micrometers and 500 micrometers. It obviously depends on what cell we're looking at. Like the yolk of a chicken egg is much larger than 500 micrometers. And it is a eukaryote cell, the biggest one you've ever seen. I doubt any of you have ever seen the yolk of an ostrich cell. That's actually the largest cell currently on the planet Earth. A prokaryote cell is about 1/10 the size of a eukaryote cell, and its size, the average size is actually 1 to 2 or 3 micrometers. 10 microliters is getting up to the largest prokaryote cell. Viruses are about 1/10 the size of a prokaryote cell, with the diameter being between 0.02 micrometers to 0.44 micrometers. And this is the diameter of the virus. You can get viruses which are larger than 0.44 microliters, but that's in their length. Like Ebola gets very close to about 1 micrometer in length, but its diameter is less than 0.044 micrometers. That's actually a megavirus, the largest virus we're going to talk about. Ebola is a little smaller, but it is longer. And the length of a virus can go down to the smallest size of a diameter and up to 1 micrometer. And you can notice that both 1 micrometer and 0.44 micrometers, that's actually the smallest size of a prokaryote cell, meaning the smallest diameter, at least of a viable prokaryote cell, is about 0.4 micrometers. Any question about any of that? So to see microbes this small, what do we need to use? Anyone? What do we use to see a
Electron
microbe?
microscope.
Actually, to see prokaryote cells, you only need a light microscope. You can use an electron microscope, but you don't need to use an electron microscope to see a prokaryote cell. To see most eukaryote cells, you also need a light microscope. Although frog eggs and the yolk of a chicken egg are eukaryote cells that you can actually see with your eyes, your naked eye. So this is showing you what you can see with your eyes, a person, the yolk of a chicken egg, the frog egg cell, and that's about the smallest thing we can see with our eyes. Can see a little bit smaller, but not much. Then everything smaller, you need a light microscope to see, and that's most eukaryote cells, all prokaryote cells. Things smaller that you can't see with a light microscope, you need to have an electron microscope to see. And that would be viruses, complex proteins like well, excuse me, complex molecules like proteins and large lipids. Small molecules like water, you can also see with an electron microscope. And then the very smallest thing you can see with an electron microscope would be an atom. And this is showing you the relative size of things. The smallest thing that we're going to talk about in this class is an atom. Atoms get together to make small molecules like water. Small molecules and atoms get together to make large molecules like lipids and proteins. Proteins and large molecules get together to make viruses. Large molecules get together to make prokaryote cells. And I can't really say prokaryote cells get together to make eukaryote cells, but we have molecules which get together to make eukaryote cells, and then eukaryote cells get together to make a person. Any question about any of that? So you don't need to know the sizes here, but you should know the relative ranking. And do realize that this graph has an arrow in it. I got the arrow right there. That's not 1 micrometer there. This is 1 micrometer here. That should be 1 millimeter here. And I don't know why the author of this textbook kept repeating this error on this graph for publication after publication, because I think this error has been repeated about 12 times. All right, any question? Not in our current version of the textbook made for Clark College, but in the current version of the textbook, this error has been repeated about 12 times. All right, here we're looking at a compound light microscope. It's a compound light microscope because it has 2 or more lenses that magnify. The magnifying lenses is the ocular lens found here and the objective lens found here. The total magnification is the magnification of the objective lens times the magnification of the ocular lens. Most of the microscopes in our lab have an objective lens of 4x, 10x, 40x, and 100x, meaning as you can sort of see 4 different lenses, 1, 2, 3, and there's the 4th there. And you can rotate this on the rotating nose piece to get the different objective lens in place. And what you do is you take like the 4x magnification and then multiply it by the magnification of the ocular lens. In the microscopes in our lab, all of the ocular lenses magnify 10 times. So the total magnification of the 4x lens is 40 times. And then the total magnification of the 10x objective lens would be 100 times, etc. Because all of our microscopes have a 10x ocular lens. Any question about any of that? Now, the best microscopes and the most expensive have a 20x ocular lens, and it magnifies 20 times. Obviously, at Clark College, we do not have the best or the most expensive microscopes available. You should know the different parts of the microscope labeled here, the ocular lens. You don't really need to know the prism, but you should know the objective lens, the tube body. This here would be the handle you hold it with. You should know we don't have it illustrated here, so I'll discuss it later. Condenser lens, that is not a magnification lens. What it does is it fixes the cone of light coming from the lamp or illuminator. So that we have a cone of light coming through the specimen, which is on the slide. And then the base of the microscope shown here. So you should know the parts of the microscope shown here. The ocular lens, also called the eyepiece, the body of the tube, which you can hold it right there. Some of them even have a hole, how do you call it, a handhold right in this area. The stage is what holds the slide in place. The stage clips, which you can see sort of right there. There it is right there. Sorry, it's right there. And I'm not seeing the stage control here. That's for moving the slide. We'll look at another microscope and see the stage controls. But the stage holds the slide. The stage clip holds the slide in place so the slide can't move, at least unless you want it to move, in which case you use the stage controls. The condenser, which focuses the light on the stage, does not magnify. The diaphragm makes the cone of light coming through the condenser. And sometimes the diaphragm is below the condenser. On some microscopes, the diaphragm is above the condenser. The diaphragm opens and shuts like a shutter to a camera to allow either more light to get through. If it's totally open, then you get a lot of light coming through. And then you can start closing that diaphragm down until you get a pinprick of light coming through. The illuminator or the lamp is the light source. Then we have the coarse focusing knob that moves the stage up and down coarsely or rapidly. And then the fine focusing knob that moves the stage up slowly and only a little bit, either up or down, for fine focusing. Any question about that? So the coarse focusing is the large knob, and then the fine focusing is the small knob. Also shown is the light switch, which you turn it up to get more light, and then you turn it down to get less light. You turn it down all the way, you turn this one off. Here is a simple microscope, the one made by Antony van Leeuwenhoek. A simple microscope only has one lens to magnify. So a simple microscope is a super duper magnifying lens. We have a few of these in the lab, but not many. And ours are more fancy than this. We don't actually stick what we want to observe on the pen here, right there. We have a stage that you want to put things you want to observe. All right, any question about the microscope? In the lab, maybe I'll show you a picture of the stage controls, because I'm not seeing it here. The stage control moves the slide this way and that way, as well as this way and that way. All right, a term you need to know with the microscope, resolution. Resolution is the ability of the lens to distinguish 2 points as individual points. A microscope with a resolving power of 0.2 micrometers can distinguish 2 points as 2 independent points as long as they are 0.2 micrometers or more away from each other. If they are closer than 0.02 micrometers, you can no longer distinguish them as 2 separate points. Let me go ahead and show you that here. So here we see 2 points, And you can distinguish these two as two separate points. But here, these are too close together. We cannot distinguish them as separate points. Any question about that? And that's because they are closer than 2 nanometers away from each other, or 0.2 micrometers. Uh, 0.02 micrometers is equal to 2 nanometers. Any questions about resolution? All right. So the best resolution we can get with white light is about 200 nanometers, or 0.2 micrometers. Anything that is closer, you cannot resolve it as two independent points, and that actually is due to the wavelength of light. So the best resolution you can get with white light is about 200 nanometers, or 0.2 micrometers. The best compound light microscopes have a total magnification of about 2,000 times. That would be the 100x objective and an ocular lens of 20 times. And that gets down to 200 nanometers. If we were to make a microscope that could magnify more than 2,000 times, it would be empty resolution, because we cannot magnify anything better than 200 nanometers away from each other, because no matter how much we magnify this, it will always be a blur. Any question about that? And the reason why 200 nanometers is the limit is because that's the limit of the properties of the wavelength of white light. Any question about any of that? So our best microscopes magnify about 2,000 times. If we were to make a better microscope that would magnify higher, like I said, it'd be empty resolution. We wouldn't be able to truly magnify. And that's because of the limitation of white light. You can't magnify more than 2,000 times and get effective magnification. Another term you need to know about with a microscope is that light refracts. That's the bending of light. Refraction is the bending of light. The refractive index is the light bending ability of a medium. Whenever you have light coming through one media like water, and then coming to another media like air, light bends, making it look like this pencil is bending there. And this is the artist's rendition, where at the surface, the pencil looks like it bends, right there. Every medium that light goes through will have a refractive index, and if that refractive index is different than the media it's going through, light will bend. The refraction of light is actually extremely important for life. Like, for example, this bird, when it sees a fish in the water, it's going to know that it looks like the fish is in this position, but it instinctively knows that although the fish looks like it's in this position, it's actually down in this position, and it knows it's in this position because of its instinct, meaning it knows that light bends at the surface of water, and so what looks like it's up closer to the surface is actually down deeper. And then the fish will put its feet down to catch this fish here. It won't put its feet down here where it looks like the fish is. Any question about any of that? So this is showing you and importance of the bending of light for the bird. And it needs to know that the fish isn't where it looks like it is because light is bent and the fish is really in this position. And when the light of this fish comes out of the water and into the air, the light bends, making the fish look like it's in this position. One advantage of using oil with a microscope is that oil has the same refractive index as glass. So when light leaves the glass slide and enters the oil, none of the light will refract, meaning none of the light will bend. And that's one advantage of using oil, because if you didn't use oil, when the light left the glass, the light would bend or refract in the air. And this is showing you why refraction of light and the use of oil helps improve the resolution. When we have oil in there, and then the light comes through the glass slide, going through the specimen right there, some of that light will directly go to the lens of the lamp being unbent, but some of the light will be bent at this angle and going through the specimen in the slide. Some of it will be bent at this angle. And this light, if it goes through oil, will not be bent, this ray, whereas the same ray over here, if it goes through air, it's going to be bent with the sample in the slide. And then when the light ray leaves the glass and enters the air, it's going to be bent again. And then when it leaves the air and enters the glass of the objective lens, it'll be bent again. Why it's bent here. This light ray, when it leaves the sample, it's going to be bent at this strong angle. And then when it leaves the air to be bent at a stronger angle, this light ray will be bent at such an angle it cannot be collected by the objective lens. And this light ray here, if you put in oil, it will be bent at a strong angle by the specimen in the slide. But when it leaves the oil, leaves the glass and enters the oil, it's not bent. And this light ray will be collected by the objective lens, meaning more light will be collected by the objective lens if you use oil than if you don't use oil. And it's because more light is collected that when we use oil, you have better resolution than if you don't use oil. It's just because you're getting less light when you're not using oil. And so the resolution will not be as good when you are using oil. And then this ray over here is bent at such a strong angle when leaving the specimen that it will not be collected by the objective lens, even in oil. And you notice this one actually is bent so strongly when it leaves the glass that it's actually bent back down into the glass. So the use of oil removes the refraction of light, allowing the objective lens to collect more light, which improves the resolution. And that's why using oil helps you view an object under the microscope. Any question about any of that? Let me state the use of oil prevents the refraction of light, and what we typically say is it reduces the refraction of light. It actually reduces it to zero, but all right, any questions about refraction or light microscopes? If not, let's move on to bright field illumination. The microscopes in the lab of microbiology at Clark College operate under bright field illumination, where the object shown here is darker than the background of the slide, which is glass, and so the light comes right through it, and that's why it looks white from all the light coming through from the lamp. And then the light is obtained by the specimen, or the light comes to the specimen, and then is collected by the objective lens and sent to the eye. And that's how the microscopes in the microbiology lab use. They are bright field illumination of a compound light microscope. That is what the microscopes in the microbiology lab use. Bright field illumination, and they are compound light microscopes. Dark field illumination, on the other hand, let me blow this up, has a lamp source here, and then there's the specimen there. And in between the lamp and the specimen, we have an opaque disc blocking any light directly leaving the lamp and coming to the specimen. And that makes the background of the slide look dark because no light is coming from the lamp to the specimen, at least directly. So the background of the slide looks dark, and then the specimen looks darker than the bright field illumination, but you can see the specimen. What happens with the specimen is some of the light comes at a weird angle coming to the specimen, and the light, if the specimen wasn't there, would actually, because it's at a strong angle, would not be collected by the objective lens, meaning the light, because the direct light is blocked by the disc, only light coming at an extreme angle coming through the specimen, and this light would not be collected by the specimen. However, the specimen can refract some of the light, and that's bend it, and so some of the light, which would be normally, if the specimen weren't there, would not be collected by the objective lens. It can be bent by the specimen so that some of the light can be collected by the specimen. And that is why the specimen, you can view it, it's not just dark. And that's because the light coming through it is bent by the specimen at a strong enough angle that some of the light will be bent so that the objective lens can view it. And that's why the specimen looks darker here than there, because there's less light being collected than in here. Any questions about dark field illumination? All right. You just need to know the general concepts of bright field illumination. That is, light directly from the lamp goes to the specimen, which is collected by the objective lens and then turned into your eye. Why the background looks light and the specimen is lighter than the specimen in dark field illumination. In dark field illumination, none of the light from the lamp directly comes through the specimen to the objective lens. And that's why the background looks dark, because none of the light coming through comes from the lamp. Only the light which comes through at a very odd angle, a very steep angle, and is bent by the specimen into the objective lens can be collected. And that's why the objective or the specimen here is darker than there, because there's less light. Any questions about any of that? If not, let's talk about an electron microscope. You can notice this is an electron microscope, that it is much bigger than a light microscope. An electron microscope uses a beam of electrons instead of a beam of light. The electrons have a shorter wavelength than light, and that allows us to magnify the wavelength of electrons greater than we can magnify the wavelength of light. Anyways, the shorter wavelength of electrons give greater resolution so that we can magnify the specimen at many times more than you can magnify the specimen with light. The electron microscope allows the examination of viruses and internal cell structures, structures which are too small to be seen by the light microscope. There are two main types of electron microscopes. There's actually 4 total types. There are 2 you need to know, the main types. You need to know transmission electron microscope, shown here. Let me blow that up. So this is a transmission electron microscope. And then a scanning electron microscope. What do you notice between the difference between transmission electron microscope and scanning electron microscope? Anyone? What's the difference between this slide and that slide? Or that slide and that slide?
It looks cut in half.
Say again?
It looks like it cut in half.
Yeah. With transmission electron microscope, you're only seeing one plane through the cell. Meaning you cut the cell and you're only looking at one plane of the cell. With the scanning electron microscope, on the other hand, you're seeing the cell in 3 dimensions. While the cell has a surface in 3 dimensions shown here. The other difference is the scanning electron microscope only sees the surface of the cell, or the surface, whatever we're looking at, and the transmission electron microscope can look at the cell internally. It's only looking at one plane through the cell, but it's looking at the internal structure of the cell in that plane. Here we're looking at a crystal and with a scanning electron microscope. And once again, you can see the crystal in 3 dimensions. And this is a transmission electron microscope. I think that's of a chloroplast. These are the membranes of the chloroplast. And you can see, once again, you don't see the object in 3D. We're only seeing the object in 2D with transmission electron microscopy. And we're only seeing one plane through this cell. There is a third type of electron microscopy. We're not going to be examined on it, so you don't really need to know about it. I'm just briefly going to talk about it. The third type of electron microscopy is called scanning tunneling microscopy. It uses a metal probe to scan a specimen. And this is the highest resolution we can get with a microscope, scanning tunneling microscopy, where you can see something as small as 1/100th the size of an atom. And here we're looking at scanning tunneling microscopy of a protein from E. coli. The fourth type of an electron microscope, once again, you're not going to be tested on this, so you don't need to know it. I'm just briefly going to talk about it, is using a metal and diamond probe inserted into the specimen, and then you can see the specimen in 3 dimensions. And this is the toxin from Clostridium perfringens, shown here. All right, any questions about the electron microscope or the difference between an electron microscope and a light microscope? Let me ask you, what's the difference between an electron microscope and a light microscope? There are 2 very easy differences.
The light microscope uses glass, and the electron microscope uses the electromagnetic lenses.
You are correct that oh, you're right. They're using a lens, and the lens is glass with a light microscope, and the lens is the electromagnetic field of the electron microscope. The main difference is, oh, that is a good one, but that's not the one I was getting at. The main difference is an electron microscope uses electrons, and the light microscope uses light. And then the other main difference is the electron microscope can magnify much smaller than the light microscope. And you are correct, by the way. So that would be like a third difference. All right, we've made preparations to look at our cells under the microscope. And here we can see our cells, but here we can see our cells also. Which cells are easier to see, the ones on the left or the ones on the right?
The right one.
Yeah, the ones on the right. The only difference between these cells and these cells is these cells have been stained by a dye. These cells are unstained. And staining allows us to much more easily see the cells because staining increases the contrast between the cell and the background. And as you can see here, there's not much difference between the cell, meaning there's not much difference in contrast between the cell and the slide or the background of the slide. And that makes these cells hard to view, whereas these cells have much deeper contrast between the cell and the background of the slide. And that is all staining does, increasing the contrast between the cell and the background of the slide, which usually the slide is just glass. Any questions about staining? All right, with bright field microscopes, that's the normal compound microscope in the lab, most microbes are colorless and have little contrast with the slide. To see them, we need to stain them. Otherwise, you won't see them very well. Staining is coloring the specimen or background to increase its visibility by increasing the contrast between the cell and the background of the slide. A smear is usually made before you view the cells. A smear is a thin film of solution of microbes on a slide. So prior to staining all cells, you have to make a smear. And with bacteria, we call it a bacterial smear. I suppose we could call it a microbial smear as well, where you put a thin layer of bacteria on the slide. And that is always done before you begin the smear. So before you begin the stain. So staining is not making the bacterial smear, and making the bacterial smear does not involve staining the cells. The smear is something you do to the cells before you begin to stain them. Usually what you do to make a bacterial smear is put on a drop of water and then add a small amount of cells to the drop of water. You then make the microbial smear and spread it over the slide. You allow that to air dry. Once it's air dried, you then heat fix the cells onto the slide. Heat fixing attaches them as well as killing many of them. Usually heat fixing is done by putting the slide either quickly through the Bunsen burner, meaning the flame of the Bunsen burner, or putting the slide near the heat of the Bechtel incinerator, and then just leaving it there for a short amount of time to heat fix the cells. Any question about any of that? You do not want to burn the cells. You actually don't want to even really heat the cells to anything beyond what you can feel and say the slide is getting hot. So once the slide starts getting warm, that's all you need to heat fix the slide. Once it gets hot, you probably heated it to the slide too much. When staining a cell, we stain the cell with a stain or a dye. These two words are used interchangeably. A stain is a salt consisting of a positive ion and a negative ion. One of the ions is colored, that is called the chromophore. A basic dye is one where the chromophore is in the cation of the ion or the positively charged molecule or atom in the stain is the one containing the color or the chromophore. And then the negative part of that molecule will not be colored. With acidic dyes, the chromophore is in the anion or the negative part of the molecule. And the positive part of the molecule is not colored and does not stain the cell. So acidic dyes, the chromophore is in the negative part of the molecule. In a basic dye, the chromophore is in the positive part of the molecule. Most dyes in microbiology are basic dyes, but we do have some that are acidic. Bacteria have a slightly negative charge. Therefore, a basic dye is better for staining the bacterial cell than a acidic dye, because the acidic dye has a negative charge, the cell has a slightly negative charge, the chromophore will be repelled from the cell. With the basic dye, the chromophore has a positive charge and it will be attracted to the bacterial cell. Most dyes in microbiology are basic dyes, like crystal violet, methylene blue, malachite green, and safranin are all basic dyes. And we'll talk about each of these dyes if you don't know them. I think we talked about crystal violet, maybe safranin. But we will talk about all these in the course. Any questions? I think we're at time to stop. Yep. All right, if there's no questions, I'm going to end it here, and I'll see you at 7 for the lab. All right, bye.